you're just fired. You can just leave now. Bye. Ciao. All right, so we are almost all. I think we're 43. We can start. I wanted first to revisit one concept from this morning. Let me just share the screen with you because I've got some questions and comments about the LTP, right? Yesterday, if you remember, we said that one important feature of the neural code or action potential, or however you want to call it in this context, is that information, like the shape of the action potential is basically the same or almost everywhere you probe it, right? It's very stereotypical. And in itself, it carries very little information, but the way, like the important message is defined by who is emitting this action potential. So who is saying the action potential and who is hearing the action potential. And I want you to just get the glimpse that the same is for LTP. LTP is a means of changing the weight of a synapse to make it stronger or less strong. The effect that it has, it depends who is the presynaptic and the postsynaptic partner, right? So in the end, LTP is the way in which memories are built, and this is how it has been proposed. But as someone was commenting today, I think it was Anna maybe about addiction, it can also underlie other interesting phenomena. It can happen between different cell types than only excitable neurons. So in the end, really the effect of the LTP itself depends on the message that is transmitted through the synapse that you are basically using, right? So yeah, I just wanted to stress this further and to not be misunderstood. One thing that I want just to remind you is that still we have a Slido open. So if you want, you can interact through that anonymously. I see that people don't do it. I mean, there is really no reason, no judgment about the question. Two things that you should have learned in this afternoon is that neurons can have very different activity patterns, which are influencing the way they connect to each other. Especially neurons have very different activity patterns when you start to throw in interneuron in the mix. That's why, because they create like different feedback type of projection and dynamics of the membrane that are able to reset depolarization that might not end up in action potential. So can we understand how neurons work together and which patterns of activity they create? And by the activity pattern that they create, can we go back to try to understand how they connect to each other? The answer is yes-ish, but we still need to record from a lot of neurons at the same time. In your network, you're recording from all the input neuron, all the output neuron, and all the interneurons. Do this with the regular physiological technique that I told you yesterday is extremely complicated, even with tetrads, because the number of neurons that you can record is quite limited. Fortunately, the past 10 years, I've seen an explosion of techniques that allow us to record from many, many neurons at the same time in order to understand how these neurons influence each other and what is the hidden message of the brain, something that we're going to look at tomorrow and Thursday with our decoding strategies. So from the physiology point of view, we have had the development of probes that instead of having only 4 sites of recording, have 920 sites of recording. This is an image from a paper that was published, I don't remember if last year or 2 years ago, but these are neuropixel probes that some of you that are interested in neuroscience might have heard about. These are long, very thin silicon probes or other materials that basically are equipped with a high density, high number of recording sites distributed at high density. And you see like one of these probes, of course, has connectors for basically like transmitting the signal that is picked up at the level of these sites to different type of boards, amplifier, and so on, so that in the end, we can record from really many, many neurons at the same time. Here you find an example from the same paper in which the basically the trace of the probe is highlighted in blue, and you see that at every one of these sites you have one different trace from which you can see that there is some sort of coordinated activity at a certain level of the brain, right? I think in this case they were recording from roughly 1,000 of neurons, which makes the analysis a little bit complicated, of course, but also simplifies it in a different way. The way in which we isolate individual neurons from this sea of spikes is always following the same principle that I told you before. Just this time, instead of having only 4 electrodes, we have a lot, like multiple sites that we can use to basically like cluster the spines together. So this is something that is extremely interesting to apply, especially because it's something that allows us to record spikes with a high temporal precision from many neurons in the brain. But I want to introduce just another concept right now, that is the idea that we can maybe try to infer neuron activity not by looking at the electrical activity that these neurons are producing during action potential, but at the way in which basically like the neuron functions from a biochemical point of view. In fact, we can image calcium dynamics during during neural activity. And what is the principle behind that? The principle is that calcium is a powerful second messenger, and many ligand and voltage-gated channels are actually permeable to it. So when you have neural activity into the cell, the intracellular concentration of calcium increases. And that can be taken as an indication of the cells being active. So if we were able to measure the dynamic change of calcium concentration in a neuron, we would be able to deduce if the neuron was active or not without recording the electrical signal. If every time a neuron fires a spike, you have a bump of calcium concentration within the cell that goes higher, you are able to infer what the neuron has been doing by just looking at the calcium concentration within the neuron, right? So this would be a way to look at activity of the cells without being able to without having to necessarily record electrical activity within the brain. So is there a way in which we can visualize calcium and calcium dynamics? Turns out that there is, because there is a lot of different calcium indicators that have been produced and synthesized, both synthetically and genetically. Calcium indicators are molecules that are able to bind calcium in a reversible manner. So they bind calcium depending on the concentration of calcium within the cell and are able to emit some fluorescence when the calcium is bound. So you have a situation in which you have the protein in a resting state, which has minimal level of fluorescence, but we'll see later how there are many different types of ways in which this can work. When calcium is added to the mix, the binding of calcium with this protein, in this case, is able to induce a conformation change in the protein that allows the protein to emit a lot of photons. So in the end, we can measure the fluorescence of the individual neurons that is given by the binding of the calcium indicator with calcium and infer the spiking activity from this type of measure. The problem here is that you see how many different type of passages there are, right? You're not really recording directly the activity of the neuron in this case, like we have done before with the electrodes. With the electrodes, we were going at the source of the way in which neurons talk to each other. We have recorded the electrical signal of the neuron. In this case, we are indirectly recording the electrical signals of the neuron by recording an indicator, which is the expression of the change in concentration of calcium within the cell. This is important because this is really the main disadvantage of calcium indicators, right? That you are deducing that the neuron is active by the way in which calcium is handled by the cell. But there are many different ways that might even give an increase in calcium without being necessarily cellular activity. So this is the main source of noise when we're going to look at calcium signal within the cell. However, if we are able to visualize and to have the cells filled with this calcium indicator, we can actually do something cool. That is to just use a microscope and basically look at the way in which calcium increases and decreases within the cell by just looking at how much fluorescence there is in this neuron. When you have an increase in fluorescence, you see that the cell was most likely active because a lot of calcium has gone in, then the cells goes back to baseline, and the calcium, of course, is handled in the way in which the calcium is handled, is stored intracellularly, is pumped up extracellularly, so the concentration decreases and the level of fluorescence goes back to baseline. In the end, when we put our, let's say, spotlight on just one neuron, we can record the change in fluorescence of the calcium indicator. So you see this trace here, this yellow trace here, and use that as an indication of the moment in which the neuron was active. This is actually something that is real. So one cell is not exactly this cell from this movie, but one cell was at the same time touched with an electrode so that you could actually record the electrical activity of the cell and indicate the spike here. And at the same time, it was followed with calcium imaging in order to measure the change in fluorescence given by the change in calcium. And you can see how there is a very nice correspondence between the moment in which the cell was active and the moment in which calcium was changing. Of course, one thing that you have to take into consideration, and that some of you might have immediately seen, you see it here. Here, the cell was firing one spike. Here, the shape was not like instantaneous. The fluorescence of the neuron was not instantaneously going down. The rise in fluorescence is very fast. So the binding of the calcium indicator to calcium is very fast. But once the calcium is bound to the calcium indicator, it takes a little bit of time for calcium to be released. So that means that there is an exponential decay function that tells you that even if the cell was active only in one moment in time, in one instant, for example, you will still continue to see that the fluorescence is not going back to baseline for a certain amount of time. The process of calculating how many action potential could actually underlie this particular deflection is called deconvolution, and we're going to see it after in a very brief way. For now, the take-home message that, one second, I finished the thought. For now, the take-home message that you have to take from this slide is that by using a calcium indicator within the cell, you can take a fluorescence trace that is not linearly, but you don't have to know this, but it still is a measure of the calcium concentration within the neuron, which is changing as a function of neural activity. Yeah, Nuno, right?
Yeah. Is it possible to visualize these dynamics in vivo?
Yeah, this is actually in vivo.
Okay.
Yeah, so I'm showing you later how you do it. So basically, the whole presentation is going to be focused on in vivo. Yeah, but you can do it also in vitro. I mean, these first were done in vitro. There is a lot of difficulty and a big road that has brought us to imaging. Yeah. So you could say, okay, I can record. Okay, the advantage here is simple, right? In one small field of view, you already have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 cells that can be recorded simultaneously. This is, even with tetrads, is actually quite a good number. So one could say, why should we say calcium imaging instead of using electrophysiology? So calcium imaging, there is a lot of positive and negative sides for both. Let's go through some of these aspects. How many neurons you can record at the same time? You can record up to thousands of neurons at the same time with calcium imaging. With the traditional electrophysiological recordings, this number was down to tens. Now, with the neuropixel probes and the silicon probes, you can actually go with thousands. The resolution that you have with the cell, meaning you know which cell is actually active, is direct for calcium imaging and indirect for electrophysiology. What I mean, yesterday, if you remember, when I told you that we were assigning spikes to cluster and thinking that one cluster was actually one neuron, but there might be contamination for which one cluster is not really one neuron, and you're not entirely sure that this is the case. With calcium imaging, you're sure that's the neuron, that's a cell body, and no other cell body can be overlaid to that neuron. We'll see how there are sources of contamination in this sentence, but mostly you can be more direct in the measurement. One thing that is very important is that because we can express calcium indicator based on a specific promoter or a specific gene that is expressed only by a population of neurons, we can record from genetically identified neurons with calcium imaging, but we really do not record with genetically identified neurons with calcium recording. We just lower our tetrads, electrodes, neuropixel probe in the tissue, and then we listen to the sea of spikes that every neuron that is close by to that particular electrode is telling us. There is a shortcut to that, involves the use of optogenetics, which is not trivial, and which we're going to talk about on Thursday. The big disadvantage of calcium imaging is that it is a slow temporal dynamic, so as a low resolution, because of this phenomenon that I've told you before, that the release of calcium from the calcium indicator actually takes some time, is not an instantaneous thing. And also, the measure of electrical activity is indirect. You're not recording action potential or extracellular spikes. You're recording dynamic of calcium, which is correlated to your neuronal activity, but is not a one-to-one relationship. One big advantage instead is that because you can use more and more powerful objective and microscope and so on, you can zoom into dendrites, individual synapses and individual spines, and you can quantify the activity of individual synapses and individual subcompartment of your neuron. Something that with electrodes is extremely difficult, if not impossible. Yeah, the other thing is that most of the times you can record for calcium imaging from the same neuron over the course of multiple days, weeks, and months, which allows you, for example, to understand how the activity of one neuron changes with time or experience. This is very limited with electrophysiology because the relative position between the electrodes and the cell can actually change when the mouse bumps his head into a wall. And that means that you cannot be sure that you record from the same neuron for a long period of time. The most important and really the game-changing advantage that calcium imaging gives for recording of cellular activity is the fact that you can single out the neurons that you're recording activity from. And that's because you can use a genetically encoded calcium indicator as a calcium buffer and drive this genetically encoded calcium indicator with specific promoter. Why would this be important? An application is something that we have been talking about since yesterday and most likely today. Excitatory and inhibitory neuron have different dynamics and different activity patterns. And this is something that you have seen today when you have played out with your networks, right? Most of the time, the inhibitory neuron might even have different property, change the way excitatory neuron work. And knowing if a neuron that is active is excitatory or inhibitory really helps you to understand what the effect of this neuron is on the whole network, right? Now, distinguishing excitatory neuron from inhibitory neuron from your electrophysiological recording is extremely difficult because the difference in the shape of the waveform of these two action potential or spikes is extremely small. For example, if you have here 3 different examples, you have the classical waveform that you would get from an excitatory neuron, a classical waveform that you would get from the inhibitory neuron, which is this black trace here, and a waveform that you would have from an en passant axon of an excitatory neuron. You see how, yes, there is tiny difference in the width of the spikes. There is a tiny difference in the duration of the whole spikes. But in general, if you plot spikes in these 3 different scenarios, you're really not able to distinguish them in a very convincing way. So most of the time you try to separate activity that comes from excitatory neuron and activity that comes from inhibitory neuron based on the waveform and on the refractory period. But in most of the case, it's very complicated. On the other hand, you know that inhibitory neurons are characterized by the expression of a lot of different markers like parvalbumin, somatostatin, VIP, calretinin, and so on and so forth. And each of these markers is actually able to identify a few subpopulation of interneurons which have different type of connectivity and different intrinsic properties. So by using just 3 markers, parvalbumin, somatostatin, and the 5-HT3 channel, you are able to single out and distinguish many subpopulation of interneuron that are basically populating and integrated into the cortical network. And by driving the expression of this interneuron, of the calcium indicator in a genetic way and based on these markers, you're able to single out activity from the parvalbumin positive neurons, which are the basket cell targeting the perisomatic region, the somatostatin positive neuron, which are targeting the dendritic region, and the VIP 5-HT3 neurons, which are targeting both the dendritic, perisomatic, and the other interneurons into cortical networks. So having a distinction of these 3 classes only with physiological method would be almost impossible. But thanks to the fact that you can actually express the calcium indicator specifically only in this population and listen only from this population, you can understand the contribution of different subpopulation of neurons, of interneurons in this case, to a specific computation of specific behavioral paradigm. This, in the last year, has been expanded enormously by the amazing work that the Allen Institute in Seattle has done to identify differences also into excited neurons. Before, excited neurons were considered to be sort of an homogeneous population in which you could distinguish different classes of neurons, but not too many. You have pyramidal cells, which are different from stellate cells, and you might have thought about this last week, so I'm not going to go into detail. But what basically the Allen Brain Atlas or the Allen Institute basically done is to characterize systematically properties of neurons that are belonging to different cortical area, cortical and subcortical area, based on the expression of their genes, on their developmental origin, on their connectivity, or their activity. And based on the property, you can identify subpopulation of excitatory neuron from which you can record selectively, for example, through calcium imaging, or whose activity you can manipulate selectively with genetic means that we will discuss about on Thursday. So in the end, you can, by selecting the population of neurons that you record from, really understand the logic by which different neuron contributes not only to the computation of the brain, which is the different way in which neurons are active in space and time, but also to the behavior that the animal is doing when those neurons are active. And this is a big advantage that calcium imaging has brought that was, I would say, impossible before 10 years ago, before optogenetics, still extremely difficult only with electrophysiological means. Okay, so this is a general principle. I hope that we are all on board on this, because this is really like one of the advantages and one of the technological discoveries or invention, if you want, that has propelled neuroscience to a different level, to understanding really not only the contribution of individual area of cortex or of the brain in general to specific behavioral task or paradigm or phenomena, but also of individual population of neurons to those same behavioral phenomena. Any question? Okay, so if that is clear, I'll move forward. So to target a specific population of neuron, what you need is a system that allows you to a specific marker in a specific population of neurons and a genetically encoded calcium indicator. So you should know by now during your course of study that the genetic toolbox to express gene in specific population of neurons is actually quite large. Right now, we have system in which we can drive the expression of protein in specific population of neurons that express all the same gene. So in this case, one of the most diffuse and the most popular way of having single cell or single subpopulation resolution for the expression of our marker reporter protein, which is in this case GFP, or a genetically encoded calcium indicator, is CRE. Have you ever heard of it? I assume that you are very familiar with the concept. So I'm not going to go into detail with that. I just want to say that the CRE-LOC system is extremely versatile and is extremely used in neuroscience to label different populations of neurons. We will see how you can drive CRE according to many different principles on Thursday, even who is being active at a certain period of the animal behavior. But this has really also become one of the two pillars for modern neuroscience, they would say contemporary neuroscience in the last 10 years. By CRE, for example, you can label differently the two populations of interneurons that I was telling before, right? That is the parvalbumin neurons and the somatostatin expressing interneurons. So in this case, you can have expression of a reporter only in the somatostatin expressing cells. Here you can have an expressor of the reporter only in the parvalbumin expressing neuron. And you see that the parvalbumin expressing cells in red here, in green here, and the somatostatin expressive cells that are in red here are not the same subpopulation of neurons, apart from few cases. The second thing, and the second big advancement for being able to label a specific population of neurons is the ability to have a calcium indicator which is genetically encoded. And the genetically encoded, most frequent genetically encoded calcium indicator is GCAMP. This is a protein created by the change of conformation of an enhancement GFP with the attachment of two different parts. One is the calmodulin module that is able to bound calcium, and the other one is the M13 spacer, which is actually toxic and creates a lot of problem. But anyway, what this protein achieves is that when calcium is bound to the calmodulin element of the protein, the GFP change its conformation and is able to actually express the fluorescent properties that we associate to a normal GFP. So in the end, at rest, without any calcium bound, GFP is basically not really fluorescent. But when calcium binds to the protein, to the GCAP, then the protein is able to emit fluorescence. By combining together a Cree driver line or virus and a genetically encoded calcium indicator like GCAP, we can visualize dynamics of different subpopulation of interneurons while animals do specific behavioral tasks. And you can see here how you can distinguish between excitotory neuron, parvalbumin expressive interneuron, somatostatin expressive interneurons, and VIP expressive interneurons. And you can reveal different dynamics for these three different subpopulations. So this is, in a nutshell, one of the biggest advantages of calcium, that is the ability to know who you're recording from, and to know it from a principle that allows you to express, for example, a Cree in a way that is selectively given by the expression of a specific genes, by a specific developmental origin, by a specific connectivity profile, or by a specific activity profile, meaning recruitment. Any question about this part? Okay, so now we move forward and we start to see how we could envision an experiment in which we actually use calcium imaging to record the activity of neurons. There are a few things that we need to take care of, and this is a different type of question that we need to ask ourselves when we want to establish an experiment of calcium imaging. One is, what is the model that you want to study? Second, which type of indicator you will use, how you deliver the indicator, which phenomenon you want to investigate, how do you plan to visualize calcium into the neurons, how do you want to process the moving, extract the fluorescent data, and how good are your data. These are sort of a template that allows me to explain to you some of different applications of calcium imaging. The first thing that we want to decide is, what is your model? So what do you want to image? Do you want to image cells in a dish, or do you want to image neurons inside the living brain while the animal is doing something? Of course, if you want to image cells in a Petri dish, you know this from other microscopy experiment that you have encountered during your previous blow courses. You just have to have the calcium indicator expressed by the cells in your Petri dish and put the Petri dish under the microscope, and that's quite easy. If you want to image calcium dynamic in an intact living brain, there is one complication, that is that you need to make the neurons within the brain accessible to your microscope. You need to create a window inside the brain so that you can actually see the activity of this neuron through the calcium dynamics while the brain is intact. And this is achieved through some sort of complicated procedures in which you basically remove part of the skull, sometimes even part of the cortex, but you remove part of the skull and you apply a piece of glass, like a glass cover slip, on top of your brain, so that that glass can basically be the transparent point that allows you to see the brain underneath. This is the standard preparation that has been done. You basically create a window toward your brain, and you are able to achieve the optical imaging into the brain, because the photons in that case are able to penetrate to the glass that you've used in a way that they wouldn't do if there was an intact skull in that area. And in this case, you can see different type of tissue within the brain while the animal is performing a task. An advancement of this is that you can now implant different lenses directly inside the brain to reach area of the brain that are located deep within the brain. So for example, if you want to record, this is usually very good if you want to record the surface of the brain, the superficial layers of cortex. If you want to go deeper, you need to implant a lens that allows you to arrive very close to the area that you want to image. The second thing is decide which indicator you want to use. There are many different types of indicators that you could use, and they all depend, almost the one that you choose depend on the type of application that you want to use it for. So in the history of, let's say, imaging calcium dynamics, there is a big divide between the chemical type of indicator that are synthesized in a lab and the one instead that are expressed as a sort of from a genetic constant. So the genetically encoded calcium indicator and the chemical calcium indicator. And also these different type of indicator work in different way. So there are different principles by which their fluorescence is increased or changed by the binding with calcium. There are bioluminescent protein in which the binding of calcium is able to release a component of the molecule that is basically fluorescent. In this case, you see that this is sort of an irreversible phenomenon most of the time because this part is not bound from the receptor anymore. There are the chemical calcium indicator in which they have a calcium chelating sites and a fluorophore like FURA2, in which the bonding of the calcium ion to the molecule induces a conformation change that makes the part of the fluorophore part to be fluorescence. There are the ones that are based on the FRET principle, that is transfer of energy between two different components of the molecule. In this case, it's always based on the cal molecule and M13 separator. When calcium bind, these two parts of the protein come together and there is transfer of energy from this part of the protein to this part of the protein, so that basically the light that is emitted at a specific wavelength. And then there is the single fluorophore genetically encoded calcium indicators like GCAMP that I was telling you before, that are based on the GFP protein. When calcium binds, GFP goes into a conformation change that allows it to be fluorescent while it's not fluorescent at the baseline level. Of all of this, most of the time you only need to remember this one. The genetically encoded calcium indicator, in particular GCAMP, is by far right now the most used and most prevalent type of genetically encoded calcium indicator that is used in neuroscience. The other thing that you want to decide before is how are you going to deliver the calcium indicator? And the way in which you deliver the calcium indicator depends on the indicator properties itself. There are indicators that cross membranes and indicators that instead are not able to cross membranes. The population that you want to study, do you want to study all the neurons in a specific area of the brain, or do you want your calcium indicator to be expressed only by a population of neurons? And the last one is your experiment. Are you doing acute imaging, meaning you do one session of imaging and then the experiment is done? Or do you want to go back and revisit the same cells over and over again over different sessions? There is different modalities by which you can deliver the calcium indicator into the neurons. You can use an electrode and inject the calcium indicator directly into the cell. You inject the calcium indicator directly to the cell in similar way, but using a patch clamp technique. Or you can do a single cell electroporation, meaning that you apply a little bit of a current so that you create pores in your membrane and allow the calcium indicator to flow in by following electrical gradient. You can do an acute loading on the network by injecting into the brain in the extracellular medium, provided, of course, that your calcium indicator is able to cross the membrane. Otherwise, it will get stuck into the extracellular membrane and cellular space and don't cross the membrane. You can try to label neuron based on their projection by using, for example, a conjugation of your calcium indicator with dextrin. Dextrin is a protein, is basically like a molecule that is picked up by axon and that travels retrogradely to the soma, so that you can label a neuron on its axonal projection. You can do a bulk electroporation, which is basically the same principle here, just this time instead of targeting one individual neuron, you target a population of neurons in the network. Or you can use the classic genetic way of expressing calcium indicator, that is by using a virus, which is injected locally into the population, into area of the brain that you want to study. by doing in utero electroporation. That is a technique that allows you to inject a dye in the brain of embryos in utero and then to apply a current to have this genetic construct being uptaken by the cells. Or you can use transgenic animals. You're gonna look at this way of creating transgenic animals and so on in the next blog course, so I'm not gonna be deep in that. I just want you to basically get the gist of the fact that there are many, many ways in which you can give the indicator to your model system. Some of those rely on the ability of the self, of the indicator to cross the membranes, all these B systems here. Some instead rely on the ability of to inject directly the dye within the cell of interest, and some of those are based on gene expression. One way that we use most prominently to deliver our calcium indicator topically in a specific network in the brain is to inject a viral vector, which has been modified so to encode the construct that is responsible for the expression of the genetically encoded calcium indicator. And we do that by producing them in vitro. This is something that Fabia is very good at doing. So if you have questions about it, please ask her either today or tomorrow during the break. And once we produce and harvest the virus that contains the gene for the expression of the genetically encoded calcium indicator, we inject the solution containing the virus topically in the area of the brain where we actually want the virus to be expressed. So all this part is to aim and get our genetically encoded calcium indicator to be expressed from a specific or a non-specific but still local population of neuron within the brain, so that then we can go with our microscope and record the activity of these cells by imaging the changing fluorescence of individual neurons. I think at this point, we can probably take a short break and reconvene for the second part of our experiment, that is actually imaging the dynamic of calcium within the cell in the second part of this lecture. Any question about this? About calcium indicators, about the way that we deliver calcium indicator and why we would like to use calcium indicator to record activity of neurons?
There are some anonymous questions.
Oh, yeah, I'll go and check them out. Okay, so the first one I think is are there any diseases associated with these feedback systems? Can the person that wrote just comment on what does he or she means about feedback system? Is it talking about the network motifs? Explain the question because I don't really understand this. But yeah, I'm sure that it can be expressed. I'll move to the next one.
It was me.
Ah, okay, David. Yeah.
Maybe some disease like epilepsy or other diseases that are associated with it.
So that I know, there is no disease that is associated specifically to one motif. I would say that it's more associated to the function of a specific neuronal population, for example. So for example, in the epilepsy one, you can have that you have a decreased inhibition. So you have a dysfunction of some inhibitor neurons that are less efficient in actually inhibiting the network, and that creates this propagation of activity. But I wouldn't say that the pathology is associated to the motif itself, but it's associated to something else. That is a dysfunction of the way in which the dendrite of the membrane of the dendrites integrates signals and stuff like that. To my knowledge, no, to the specific motif. Also because the study of the brain network based on motif is actually quite recent. So yeah, I don't think that that's the case. Yeah, that's the short answer. The other question was, is expressing an artificial calcium binding protein affect the physiology or excitability of the cell? The answer is yes. So by the moment in which because it binds calcium, a calcium binding protein is a calcium buffer. And calcium has an important role into the cell. You might have had actually experience already with this in other courses. calcium-induced changing in gene expression. Calcium is used in neurons to open specific type of channels. So by definition, buffering calcium has an effect on the cell physiology. And this becomes particularly evident when you express this genetically encoded calcium indicator for a very long time. The cell starts to be not healthy anymore, and after a certain period of time, you actually cannot record the activity of this neuron because it might even die. This is a well-known bug. Most of the times, the calcium, the genetically encoded calcium indicator expression stabilizes to a steady state, but some neurons express too much, they become sick, and they actually die. One thing that is actually toxic in association to the calcium indicator is the virus itself. The type of virus that you use might actually harm the cell on its own. Of course, these are modified viruses, which have no virulence and they cannot replicate and stuff like that, but still they use the machinery of the cell to produce some proteins. And the other thing is the M13 elements is actually toxic to the neuron. So this is something overlooked when we talk about GCAM, but that fragment by itself is actually toxic. Besides calcium, is there any other component of cell physiology that could be visualized in vivo? Yes, absolutely. And people are developing indicators as we speak. Now there is the new frontier. So first of all, there is let's say the biggest promise for calcium imaging or for in vivo imaging in general is using dyes that are able to report the membrane voltage. These are the voltage indicators. And these are basically like extremely powerful because I told you that the calcium indicator shows you the activity of a neuron in an indirect way, but these are able to tell you the activity of the neuron in a direct way, right? The problem with that so far is that it's so fast, the dye, that it's very little, the signal, it's very fast, and there is a very little deflection from the baseline. I mean, there is a lot of noise, but people are trying to optimize those. But right now we have indicator for serotonin, we have the indicator for glutamate, so we have indicator for neurotransmitters, we have indicator for neuromodulators, we have indicators for reduced proteins. So for example, you can monitor and visualize the redox state of the cell. The principle is being applied to many processes of the cell itself, not only neurons. Everything that you can attach a sensor to, you can basically get an indicator and use it for imaging. Is the virus harmful for the cell's mice? For the cells, in some cases, yes. And that's what I've said before. It depends on the type of virus that you use. What I showed you before, these viruses here, these are adeno-associated viruses. They're not very toxic, neither for us or for neurons. But there are other viruses that are used for their specific infection cycle, like rabies viruses, for example. And those are very toxic for cells. So they hijack the intracellular machinery for protein expression. The cell will suffer by the fact that it cannot really translate its own protein. And they're sort of ticking bombs. And the moment in which they infect neurons, the neuron has few days to live. But still, one can use these viruses in an effective way to do experiment in system neuroscience in a way that is actually meaningful and not too harmful for your system. At the same time, one thing that I want to pass is that almost every type of manipulation that involves dipping an electrode into the brain, creating a window into the brain, aspirating part of the tissue to see the hippocampus, implanting a lens within the brain, will create a certain amount of damage. And one always needs to carefully consider that and to basically like make sure and control the damage in itself is not responsible of the type of activity, dynamics, phenomena that is observing. And that's why one of your biggest work or your biggest, like one of the most important work that you will have when you will become scientists and researchers is to plan adequate controls. Always controls. You don't want to base your theories on artifacts that are given by your technologies. You don't want to base your theories on flukes that are given by random and spurious observation. Always do the appropriate control. It's one of the biggest part of setting up a proper experimental project. All right. So, any other question? Okay, so if there is no other question, there is a small part to do. We can go on we can have a 15 minutes break and see you back here at 5. How about that? Okay, so I'll take a coffee. See you in a bit.